Okay. Okay. Hello, everybody. We can start now. We wait for a couple of minutes, and I announce a couple of notes before everybody join us. So we have the chat box. I enabled the chat box. Can you tell me if you hear me well and the quality of the camera and the image is good and the video is good? On the chat box on the right hand side. Okay, that's great. So as you can see, we have the new platform. Thank you so much. Yes, sounds great. All right, thank you. So we have a new platform. We kind of transfer from GoToWebinar to Crowdcast. So we are just checking if this platform is great for our seminar series. And thanks to our sponsor, Montreal TransMedTech Institute, to provide this opportunity for us to use this Crowdcast platform, which is a web-based platform. So there are so try to familiarize yourself with the platform. There on the bottom you have there is a box of ask a question, Q&A box. You can ask your question, and the good thing about this platform that the question can get vote, and people can vote to the best question, and the best question go to the first rank. So and we get the priority and to ask that question. And then there is a poll option that we will ask you in the middle of the event after the presentation to take this poll and tell us about your opinion about the today talk and the organization. And so that's it. So let's start off today's seminar, which is very great, and I'm very excited about it. Before that, I have some housekeeping notes, and then so I'll try to share my screen again, and then okay. So we got the permission from our great speaker today to record his talk, and then as usual we put the recording on YouTube channel. And so go and subscribe to our YouTube channel, and if you want a refresher and you want to watch it again, you can do it. And also for the people that cannot attend, they can go always to our YouTube channel and watch the videos. And also, as I mentioned, you can write your question to Q&A box, and we will send a survey after the event. So please take the survey and let us know if you like the seminar. And in the middle of this event, also please take the polls that I posted. So I will announce it later. So our next e-seminar is with Dr. Jeff Karp, Professor Jeff Karp from Harvard Medical School. He is a serial entrepreneur, and I attended his talk many times, and whenever I attended his talk, I got inspired by his insights and visions about the technology and translational biomedical engineering. He will talk about the tools for accelerated medical innovations. So we are on social media, so you can follow us, follow our Twitter account at TransBiomed, and then you can always send us your questions to each of us. And here I would like to thank the support and support from Montreal TransMedTech Institute. This institute aims to support the development of innovative medical technologies, train the next generation of professionals, and make innovations in life sciences and engineering a source of wealth for society. And their approach is based on Living Lab. TransMedTech provides an integrated environment that supports interdisciplinary collaborative processes and co-creation of new medical technologies and interventions to catalyze their development and adoption by users. If you need more information about TransMedTech, you can always check their website. I just put it there. And just quick note that I'm also a research chair in 3D bioprinting and regenerative medicine in Montreal TransMedTech Institute. So you can always also ask your question from me if you have any. And we would also like to thank our other sponsors, MedTech Talent Accelerator. The MedTech Talent Accelerator is a training program designed to launch the next generation of industry-ready talent for the Canadian medical technology sector. The program is a collaborative program between Ryerson, McGill, Biomedical Zone, and MedTech Canada and its member companies. So if you need more information about MedTech Talent Accelerator, you can always check their website. So by that, okay. So by that, I hope that you can see me. I invite our speaker on the screen so I can introduce him and he can, yes. Hi, Dr. Kazemzadeh, how are you? So you are muted. You are muted. So yes, so that's great. Yes, now you are good. I can hear you. So okay. Yeah, let's start because we got audience here, and then it's a good time to start off. I would like to introduce. This is a great pleasure and honor to actually host my colleague and friend, Dr. Kazemzadeh, today. Dr. Kazemzadeh is a biomaterials engineer with academia, startup, industry, and regulatory experience. He currently works as a lead scientific reviewer and consultant for US Food and Drug Administration, Office of Device Evaluation, Center for Devices and Radiological Health, Division of Orthopedic Devices. Before FDA, Mehdi was a supervisor research engineer at Cook Biotech company, work on soft tissue grafts. Mehdi also involved in a startup company in Canada with a focus on developing bone glues. His academic background includes postdoc at Harvard Medical School with research topic on bone tissue engineering, tissue adhesives, and a PhD in biomedical engineering from the University of British Columbia on antimicrobial peptide coatings for orthopedic devices. So far, Dr. Kazemzadeh has published about 30 journal articles with over 1,600 citations. So we are looking forward to your presentation. If you could also please share your screen, and then we can go ahead.
Sure. Can you see my video and audio is clear?
Yes, everything is clear.
All right. Great. Thank you, Human, for the nice introduction, and thanks everyone for attending. And congratulations, Human, you have lined up great scholars, and it's a pleasure to be invited on this talk. As Human said, I got my PhD from University of British Columbia in 2013. It took me 5 years to receive my PhD, and with a couple of journal articles I published. And then after, I joined a startup company in Halifax, working on a special bone adhesive for craniomaxillofacial applications. After a year and a half working on that startup, I moved on to Harvard Medical School, did a postdoc, and then after my postdoc, I joined industry in Indiana at Cook Biotech company. Well, it was great because it was a GMP environment, and I never worked in the big industry before, so it was a great experience. After working in Cook Biotech, I joined a regulatory agency, FDA, in Silver Spring, Maryland. So I've been working there for almost 5 years now. So the whole journey was great. I had academia background, and then I had a startup. I got to know startup challenges. Then I work in industry in the GMP environment, and eventually I started working in the regulatory agency. So by saying that, here is my disclaimer. This presentation reflects the views of myself.
Mehdi, Dr. Kazemzadeh, we don't see your slides. Can you just share your screen?
You don't see any of the slides? Okay.
No. So maybe you can start from the beginning of your slides, and then, I mean, you have a great long journey, but we didn't see your slides. So. Yes, I think, yes, if you go to the presentation mode, that's great.
Okay.
And yes,
this was my first slide. I didn't move on.
That's great. So yeah, so I don't know, in case we want to record, we can record from the beginning. That's great. Okay.
Great. Okay. So you already talked about my journey, and so I've been working at FDA for 5 years. In this talk, actually, I'm not going to focus on the commercialization. We had great talks so far about the commercialization, and entrepreneurs talk about their experience of translating their products and medical devices. And this talk is more focused on the regulatory overview. I'm providing you with a high level of the regulatory and the way FDA thinks about medical device. Here is my disclaimer. This presentation reflects the views of myself and should not be construed to represent FDA views or policies. So the first slide I chose is a slide on innovation versus creativity. I was always challenging by understanding this when I was in the school. So the creativity is basically thinking up new things, unleash your brain, your mind, and conceive new ideas, and just think about anything you want without any limitation. That's creativity. So basically, when you are in the school, you think up a new idea, you propose, you write a proposal on your idea, and you have a lab with limited facility, and you conduct your research, and usually the outcome would be a paper, a journal article. The better the research article, the better the publication profile would be. So innovation is different. Innovation, creativity is a part of innovation. Of course, we should have an idea to work on, but idea is not everything. In innovation, you should have a marketing plan. You have basically, you should secure your idea. You should have a quality system in place. You should have developmental strategy. You should consider all the regulatory compliances, and at the same time, you should, after you launch your product, you should have the market and so monitoring. So basically, creativity is spending money to generate ideas, but innovation is spending ideas to generate money. So in innovation, we don't think up, we make that happen. So that's the difference between creativity and innovation. Not all creative ideas are capable of being innovative. So by saying that, I get to the regulatory science meaning. In the United States, regulatory science is the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of all food and drug. So if you consider FDA as a mountain, that has a summit, that the summit would be achieving the safety and effectiveness of your product or medical device, climbing this mountain, reach to the summit, needs some tools. Those tools are regulatory science, such as standards, recognized standards by FDA, such as the guidance documents written by FDA, or advices you get from FDA. All these tools can help you to get to the safety and effectiveness goal. So again, in the process of commercialization, as you start from the discovery and ideation, you have to make sure that your patent is in place. There is no IP blocking your idea, and then you start the goal that you start prototyping and proving the concept of your idea, and you have to do some preclinical testing, usually bench testing, animal study, and some of these medical devices products would need clinical trials, and so you go through the critical clinical trials. But throughout all this process, you need to have interaction with regulatory agency. So to better understand that, I try to make examples. For example, you do some bench testing and you do some biocompatibility testing on your, say, biomaterials, and you want to make sure if those testings are in compliance with FDA. When I was in the school, I was just doing the cytotoxicity and MTTSA and maybe just a live and dead study, and then when the cells were happy, I would say, okay, this material is biocompatible, and this is not the FDA requirements. FDA, for example, for biocompatibility, will ask you to follow the ISO 10993-1 standard, which is a recognized standard by FDA, and then ask you to follow those biocompatibility endpoints. And by saying those endpoints, it includes cytotoxicity, sensitization, irritation, acute systemic toxicity, chronic and subchronic toxicity, genotoxicity, local toxicity in terms of implantation, and genotoxicity and carcinogenicity, etc. So you see all these endpoints have to follow that standards, and you cannot just do them in your GLP, in your basic laboratory you have in the school. It should be done in the GLP environment. It means a good laboratory practice. So that means that this, if you do the testing in the lab, in your lab, and send it to FDA, FDA doesn't accept it. It should be GLP, and not all testing should be GLP, but specific, usually testing related to safety, such as animal study and biocompatibility are those that should be done under GLP. So the bench testing, you do bench testing, you do other preclinical testing, and then you interact with FDA in terms of meeting the requirements, and then for the clinical trial, if needed, you have to talk to FDA to get approval for the IDE that I will go through later. And then doing all these, you are ready to submit your application to FDA for review. And it is important to start interacting with the agency as early as possible, even during the research, and having developmental stage during the developmental stage, because imagine you change some material in the middle of the preclinical testing. Then you don't let FDA know, or you don't get the feedback from FDA. Then you spend a lot of money on biocomp and animal study, etc. And after you submit, you figure out that when the biocompatibility, for example, had been done on the previous version of the device, not the device with the new materials. That's a big problem, because FDA probably would ask you to redo all those expensive testing. So it is very important to be in a interaction, to have an interaction with FDA and get the feedback on a regular basis. So after you get approval or clearance, you launch your product, and you started doing the post-market monitoring and surveillance, and that's another part of the process. So here is the FDA structure. FDA is a big organization with different centers, with different offices. The three major offices that involve reviewing the medical devices and medical products include the Center for Biologics and Evaluation and Research. We call it CBER. CBER is responsible for reviewing the biologics. Center for Devices and Radiological Health, which is responsible for reviewing the devices. Center for Drug Evaluation and Research that review the drug. So CDRH mission is to review the devices, and the mission is to protect and promote the public health. So the CDRH mission is to support the patients and providers, and basically support the sponsors or providers with a timely manner in terms of the reviewing their materials and their application submissions. So if you send your submission to FDA in a timely manner, we tell you that what our feedback would be, or if it is at the last stage, we would tell you if the device is cleared or approved or rejected. So it is important to know that the CDRH mission is to support you. It is not against us, the industry, but at the same time, we have to make sure that we take care of the safety and effectiveness of the devices. So the CDRH is consisting of different offices. Two major offices are Office of Product Evaluation and Quality, OPAC, which assures patients have access to high quality, safe and effective products throughout the total product life cycle. Before, the name of this office was ODE, Office of Device Evaluation, which was basically focused on the pre-market review of the devices. But now it has changed to OPAC, and it covers both pre-market and post-market throughout the total product life cycle. So the major programs administered by OPAC are different submissions here that I will go through: 510K, pre-market notification; Denovo classification, pre-market approval application; humanitarian device exemption, HDEs; investigational device exemption, IDEs; and pre-submissions, QSubs. So I have to say that my focus on this talk is device review. We are not going to drug or biologics. Therefore, and it's a high-level review of devices. Therefore, for biologics and drugs, please refer to the FDA website. So the OPAC immediate office, it has 9 offices under it, OHT 1 to OHT 7. Each of these offices are taking care of a specific type of devices. OHT 1 is responsible for reviewing the ophthalmic and anesthesia, respiratory, ENT, and dental devices. OHT 2 is reviewing cardiovascular devices. OHT 3, gastrorenal, OBGYN, general hospital, and urology devices. OHT 4 is surgical and infection control devices. OHT 5 is neurological and physical medicine devices. OHT 6, where I am, is Office of Orthopedic Devices. OHT 7 is in vitro diagnostic and radiological health. And we have the Office of Clinical and Evidence and Analysis and Office of Regulatory Programs. So when you send your submission based on the relevance goes to any of these OHTs for review. Another office under CDRH is Office of Science and Engineering Laboratories, Osel. So Osel is basically a research office under CDRH, which is taking care of supporting the regulatory decisions and provide research activities, including the following mission: to ensure readiness for emerging and innovative medical technologies, develop appropriate evaluation strategies and testing standards, create accessible and understandable public health information, deliver timely and accurate decisions for products across their life cycle. And also, OSEL has different divisions based on different kind of skills. So now we start on defining the medical devices and explaining different kind of classification and different kind of reviews and submissions for medical devices. So by definition, this is a lengthy text, but I couldn't change it because this is what is written in the law. Based on the Section 201 of Federal Food and Drug and Cosmetic Act, the term device means an instrument, apparatus, implement, machine, contrivance, implant, in vitro reagent, or other similar or related article, including any component, part, or accessory which has the following properties. In summary, the device should not achieve its primary intended purpose through chemical action within or on the body of man or other animals, and should not achieving this intended use should not be based on metabolizing or achieving of any of its primary intended purposes. So no metabolization or chemical interaction should happen in medical device for achieving the primary intended use. This is important to know, because if it happens, then most likely your device would be considered as a combination product between drug, biologics, or device. So what is a drug? The drug definition is an article recognized in the official United States Pharmacopoeia, official homeopathic pharmacopoeia of the United States, or official national formulary, or any supplement to any of them. Articles intended for use in diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animals, and articles other than food intended to affect the structure or any function of the body of man or other animals, and articles intended for use as a component of any article specified in clause A, B, or C. So I would say that the major part of the drugs are based on the manufacturing is based on the chemical interaction. So have this magic word chemical for drugs on mind. The other one is biological products. The biological products means a virus, therapeutic, serum, toxin, antitoxin, vaccine, blood component, or derivative, allergenic product, protein, except any chemically synthesized polypeptide, or analogous product, or arsphenamine, or derivative of arsphenamine, or any other trivalent organic arsenic compound. So the biological products, the difference between biological products and drugs basically is based on their manufacturing process, which is for drugs is chemically based, and for the biologics are biological based. When the sponsors send the biological product submission or application, we call it BLA, is a biologic license application. And for the drug, it is called NDA, or new drug application. So these are different from devices. We are not going to explain about these two products. Other complicated product is a combination product. Basically, the combination product is the combination of device, drug, and biologics. When two or more of these regulated components are mixed, then we have a combination products. So it is important, again, to make sure that the primary mode of action of your device, what the primary mode of action of your device is, because when your device is identified as a combination product, the lead center in the FDA that review your material would be the center that is involved related to your primary mode of action. If the primary mode of action of your device, your combination product, is device, then the CDRH would be the lead office. Other than that would be either CBER or CDER. So say you have a device. I just try to give examples to make it more clear. And your device has different primary mode of action. Say you have devised a new coating, and your coating is antimicrobial. And your primary mode of action is to prevent biofilm formation via chemical or biological interaction. If your coating kills the bacteria through chemical interaction, then your coating would be a combination product because it is a chemical interaction. If your coating kills the bacteria through a physical interaction, you say my coating is very hydrophobic and the bacteria doesn't like it, don't attach to it, it's a kind of anti-fouling. Or you can say, the topography of the surface is in a way that upon contact of the bacteria dies, or it kills the bacteria. It is all physical. So if you can justify FDA that the primary mode of action is physical only, then most likely your device is a device. If it is based on chemical, then most likely your product would be a combination product. So what does your device do? There are two terms at FDA that are very important in terms of devices: the intended use and indication for use. The intended use is a general purpose of the device or its function and encompasses the indication for use. It means it is an umbrella above the indication for use. It is a general purpose, saying, for example, if you have a bone plate, you would say it is for internal fixation of bones. That would be a general purpose, as an example. Indication for use would be very particular, very specific to describes the disease or condition the device will diagnose, treat, prevent, cure, or mitigate, including a description of the patient population for which the device is intended. Basically, you can say this plating system is indicated, for example, for fracture fixation or reconstruction or replantation of reduction of small bones, etc. So you are specific about the indications. And so when you submit, you should have both intended use and indication for use. In FDA, we call it IFU. Regarding the classification of devices, FDA classifies devices on three different classifications: Class 1, Class 2, Class 3, and it is based on the risk that they exhibit regarding safety and effectiveness. It is based on the probable benefits against any probable risk. Therefore, if the risk of your device is low, we call it Class 1. And for Class 1, you just need the general controls to demonstrate the safety and effectiveness of the device. I will talk about what general controls is in the next slide. The Class 2 are medium risk devices. Basically, for these kind of devices, you need general controls and the special controls. Again, for the special controls, I will talk later. Class 3 are high-risk devices. It means that general controls and the special controls are not sufficient to demonstrate safety and effectiveness of the device. Therefore, these devices are the most stringent kind of devices. It is the most time-consuming and expensive sort of devices, and they are called pre-market approval. Class 1 devices are low-risk devices. Typically, some of these devices are exempted, and you don't even need to submit a pre-market review for these devices. General controls are sufficient to provide a reasonable assurance of safety and effectiveness of Class 1 devices. General controls includes adulteration, misbranding, banned, restricted devices, registration and listing requirements, quality system regulation, and good manufacturing practices. And even some of these Class 1 are exempt from the GMP as well. An example of these Class 1 devices are, for example, the surgery or orthopedic surgery instruments are general use. Or, for example, the adhesive bandages or sunglasses or IV stands. These are Class 1 devices with low risk. Class 2 devices are moderate risk devices. For these devices, general controls alone are not sufficient for a reasonable assurance of safety and effectiveness, and for which there is sufficient information to establish special controls to provide such assurance. Usually, Class 2 devices are what we know as 510K or pre-market notification. So, special controls means that you have to follow the special controls such as performance testing, animal bench testing, biocompatibility, packaging, stability, shelf life, sterilization, reprocessing, and all these special controls usually have a guidance document that you can follow. And if you show that your device pass the general and special controls, then your device actually can be cleared. An example of these devices are, for example, such as covered wheelchair or syringe, surgical mask, or some of the orthopedic joint implants. Class 3 devices, these are high-risk devices. Basically, general and special controls are not sufficient for reviewing these devices or to demonstrate substantial equivalence. These devices are called pre-market approval, class 3, as I said, the most stringent, time-consuming and expensive sort of review. And determination of reasonable assurance of safety and effectiveness is based on a valid scientific evidence. Pre-market evaluation of quality system regulation typically conducted, including pre-approval inspection. These devices are usually high risk, and such as cervical disc replacement, such as heart valves, such as implantable neuromuscular stimulator, etc. So another thing is to note is that intended use of your device can change your risk classification. Here I have an example. Imagine this stocking that is intended to prevent the pooling of blood in the legs. It has a specific intended use. It is considered class 2 with this intended use. But the same as stocking, if the intended use is general medical purpose, it would be considered as class 1 and is considered exempt from 510K. And they have different product code. The same device, different intended use, different classification. Therefore, if you have a device that have the same a cleared intended use, it doesn't mean that it is cleared for other intended use. So how do you know the classification of your device? There is a program at FDA, it's called 513G. It's when you submit a 513G, you actually ask FDA to provide you with the FDA view about classification and regulatory requirements of your device. Your submission should include device overview, proposed intended use and indication for use, and proposed classification and regulation. So FDA feedback would tell you what classification your device has. Pre-market notification. So this is basically a device that we know as substantial equivalence subject. Basically, this is a usually class 2 devices. And the other name for 510K is pre-market notification. This is based on substantial equivalence of your device with another device that already been cleared in the market. We call it predicate. So you basically say, my device is substantial equivalence in terms of safety and effectiveness with the predicate in the market. A 510K decision making is based on first, the predicate is a legally marketed device. Your device has the same intended use as a predicate, and if your device has the same technological characteristic, if it doesn't have the same technological characteristic, then you have provided enough justification, testing, or special controls to show that the device is not raising questions in terms of safety and effectiveness. The 510K is like comparing apple to apple. It's you have a device that is in the market already, and you show that in terms of safety and effectiveness, you are substantially equivalent to that device. The FDA takes about 90 calendar days to review these kind of submission. and they are usually class 2, as I said. So what happens if your device does not have a predicate in the market, but you think it is low or to moderate risk? Then there is another classification that what we call is Denovo classification. In this classification, you have to show that the probable risk to health for the device is less than the probable benefit of the device. Basically, the general, you have to provide a special controls in addition to the general control. And basically, you say that the probable benefits of the device are larger than probable risk. But then the FDA will review the material and tell you about the probable risk, the probable benefits that you have identified are acceptable or not, or the Denovo classification is the right path if you have in advance contacted the FDA through the 513G process. So it is highly recommended to contact FDA before submitting your submission through either of these classification to make sure that you are on the right path. So Denovo is right comparing to orange and apple. That you don't have a predicate exactly like yours, but you request to classify a new device into class 1 or class 2, and you think your device is not a class 3, is not a high risk. So it is for devices without predicate, with new or new technology that introduces new different questions of safety and efficacy. Demonstration of reasonable assurance of safety and effectiveness usually require a standalone data, and it takes longer for FDA to review these kind of submissions. It's about 150 days. Another device review is pre-market approval. Is as I said, the most stringent one. Usually clinical data is required for PMAs. Clinical data may be asked for the 510K and Denovo as well, but usually for 510K we don't ask for clinical data. Denovo may require clinical data, but pre-market approval most likely always require clinical data. For this kind of review, a GMP review and inspection is required. A summary of safety and effectiveness data is prepared and made publicly available. The PMA, demonstration of reasonable assurance of safety and effectiveness, need a standalone data to address risk. Basically, there is no predicate and in the market, and your device is high risk. It is only applied to class 3 devices, and it takes 180 calendar days for FDA to review the PMA applications. So we talk about the major medical device applications and submissions. As I said, 510K is about substantial equivalence. PMA is about safety and effectiveness. Denovo, you have to demonstrate again safety and effectiveness. And 513G is a way to request for classification and identification. 510K is usually class 2, PMA class 3. And Denovo is based on class 3 to class reclassification from high risk to low risk, class 3 to class 2 or 1. So as you see, the prices that FDA charge you for these submissions are very different. So if your device is a 510K, it for a small company just costs $2,738. For the PMA, this amount is $340,000, $40,995. The Denovo is $299, and for a small company, $25,575. So you see that it is very important to know the classification of your device, because it can be a huge burden in terms of money and time for the company if your device is 510K or PMA or Denovo, and if the classification is not considered. So I can give you an example to understand it better. Imagine you have come up with a calcium phosphate coating, or calcium phosphate, say, bone graft. And you see that the same calcium phosphate bone graft is already in the market and it's been cleared. You see that your calcium phosphate has minor differences with the one that is in the market in terms of, for example, the grain size or the calcium phosphate phase, etc. And you think you can justify that, or you can show that they are not raising the safety and effectiveness questions. Then you perform the testing, you submit it to the FDA, and you have the predicate, and you justify and or demonstrate that enough controls has been conducted to show that the device is as safe and as effective as predicate. So that would be a 510K, costs you very little. Just imagine now you are becoming a little fancy, you want to add some agent to it, some bone morphogenic protein or some sort of growth factor to it. Now your device is a combination product because it has a growth factor in it, and the combination product has to be evaluated by two centers, biologics or drugs and devices. Therefore, that would increase the cost, increase the time, and change the classification. It might be a de novo if you, for example, add just a simple antibiotic to it. Therefore, you can see that a little change in the formulation can change the classification of device significantly. So heads up on that. If you need a clinical data for your submission, you need to provide, get an approval from FDA for IDE, investigational device exemption. So for the IDE, either your device is non-significant. If the device is non-significant, the IRB approval is sufficient, which is basically institutional review board approval. If the risk is significant, then both FDA and IRB approval is required. You cannot start your clinical data without having the IDE approved. So it is another kind of application that is used for unapproved, uncleared devices with significant risk, approved, cleared devices to study new indication, use on human subjects to collect safety and effectiveness data, protect, and is essential to protect the human subject. FDA timeline to review the IDE is 30 days. So another submission that is I want to draw your attention to is very important, and luckily it is free. It is FDA doesn't charge you for that, is pre-submissions. So pre-submissions are mechanism for requesting FDA feedback prior to pre-market device submission. It is valuable resource for both medical device applicants and FDA. So basically in the pre-submission, you ask FDA you can ask FDA from any stage of your commercialization, from early stage in the research to developmental stage or the clinical stage. So basically you can ask questions about, for example, animal study. You have an animal study protocol. You are not sure if it is in accordance with FDA requirements. You send the animal study protocol to FDA, and FDA review the material and get it back to you with the feedback. It is free, and you get numerous valuable feedback on your animal study. You have bench testing, you have biocompatibility questions. You can interact with FDA through pre-submission. There is a pre-submission guidance that I highly recommend you to refer to and read the guidance to understand how to submit this kind of QSubs. Another name for pre-submissions is QSub. So purpose of pre-submissions is introduction to a novel device or technology, design of a clinical protocol, discussion of a specific preclinical data or testing approach, discussion of upcoming submission. So there are good and bad questions in the pre-submission question. in the pre-submission. The good question would be like, does my device require clinical validation? A bad question is, if I win this trial, will my device be approved? We cannot say that without reviewing the whole package. Will this alternative to clinical data be acceptable? For example, say you have a real-world evidence data and you want to see if the real-world evidence can replace the clinical data. That would be a good question to ask FDA. The bad question would be, do the results from these test reports look good to FDA? You have to be specific about these questions. You can ask, like, does this standard is this a standard recognized by FDA, or do you need these kind of testing, bench testing for this device or not, or about the animal study testing protocol? These questions should be specific, so you get good answer, and feedbacks are really valuable. There are educational resources at FDA that I highly recommend you to refer to. Please look at the CDRH there, Device Advice, and Division of Industry and Consumer Education, DICE. There are different ways you can approach FDA, and it's very important to, again, I cannot emphasize enough to interact with FDA early in your process, to make sure you are on the right path, because it can be a high burden on your company if you are late on interacting with FDA, and it can be very expensive and time-consuming. So by this, I have concluded my presentation, and if you have questions, please jump in.
Thank you so much, Dr. Kazemzadeh, for the informative talk. I mean, I really enjoyed the information, and also all these steps that we need to take through commercialization and validation of our devices. So before we go to Q&A, I would like to also make some notes here, that we have a poll on the bottom of the screen, and you can take the poll. Please go and check. And if you have any question for Dr. Kazemzadeh, please write your question, and the platform gives the opportunity to people vote for your question to comes up and to the first rank or higher rank, so we can ask that questions. So the other thing that I would like to share my screen. And so, as I mentioned, our next talk is with Professor Jeff Karp from Harvard Medical School. He will talk about tools for accelerated medical innovation, and he's a professor at Harvard Medical School and serial entrepreneur. And so please make sure that register for his talk. And I attended his talk many times before, and I was really inspired by his talk. So I would like to also thank our sponsors, Montreal TransMedTech Institute. If you need more information about Montreal TransMedTech, you could go to their website. I'm also affiliated to this institute. I'm a research chair in 3D bioprinting and regenerative medicine in Montreal TransMedTech Institute. If you have any question, you can ask me, or go to their website and ask your question directly. And we would like to also thank MedTech Talent Accelerator to sponsor this talk. And for more information, you can go to their website. So by that, I would like to start the Q&A. Without further ado, we will go to the first question that got the most vote here. So, Dr. Kazemzadeh, they're asking, would you please explain how a Canadian company can get FDA approval for a medical device? Is it easier to register the company in the US for FDA?
Well, to my understanding, we get the submissions from all over the world, so it is not necessary to locate in the states to get FDA approval or FDA clearance. But again, I'm on the pre-market side mostly. Please consult with the FDA through the DICE. You will see the slides after. But to my understanding, you don't need to be situated in the United States.
Okay, that's great. Thank you so much for the answer. And the next question is that please give us more information about current regulations or updates on 3D bioprinting and 3D printing for biomedical applications.
Yes, there is a guidance document on 3D printing and additive manufacturing, what we call at FDA. So please look up for the additive manufacturing guidance document from FDA. It is a comprehensive guidance document that tells you what to do and what kind of steps you have to follow. But usually the critical steps for the additive manufacturing would be the validation and the bench testing, because I'm in orthopedic and I see a lot of additive manufacturing devices coming. So you have to, in terms of substantial equivalence, you have to show that usually comparing to molding, your additive manufacturing is not raising questions about safety and effectiveness. But I highly recommend you to look at the guidance documents from FDA on additive manufacturing.
Great. Thank you so much. The next question is that for a new technology development with different medical application, is it necessary to apply for each different medical device application separately to FDA or to apply for a broader medical usage with multiple applications?
I didn't get the question thoroughly, but if your question is about intended use, are you asking if you have one medical device for different intended use? If that's the case, then yes, every intended use is reviewed different and raise different questions in terms of safety and effectiveness, and they have to be considered. For example, if you have a bone graft that you use for dental application, that bone graft for orthopedic application should be reviewed separately.
Great, thank you so much. The next question would be, can you give some insight? I know that you are in orthopedic, but this is maybe out of your expertise. Can you give some insight on AI regulation and FDA approval?
AI regulation, I know, again, I guess we have a guidance document on that. I'm kind of ignorant on AI application, never been in that area, but I can recommend to look at the guidance document on that. Yeah,
I understand. These are really like specialized, field, so you have to go and check.
FDA started actually making progress in AI applications, but that's not my field. But I know we have a guidance document. Either it is in progress or it has been drafted.
Great, thank you so much. The next question is, with 510K submissions, do you need to perform the exact same benchtop tests as the predicate with the same requirements, or can they be varied with justification?
All of them. So it depends. You can even you can even provide justification. It depends on the severity of a risk or the risk or questions we have. If the risk is not can be justified by either literature review, the literature review may work too. If usually for 510K side-by-side comparison with the predicate is the best approach, but we also accept the acceptance criteria. If you have a standard that has an acceptance criteria on that specific testing, then that acceptance criteria may work. If there is no acceptance criteria, side-by-side testing is a good approach. And still, you can try to justify it based on literature review. And yeah.
That's great. Thank you so much. The next question is the robotic surgery considered as a medical device? What is the pathway to get approval for robotic surgery?
Yes, the robotic surgeries are devices, but again, they can have different classification based on the risk they have: class 1, class 2, class 3. And it depends on what are these robotic devices on. We get a lot of navigator system in orthopedic, which are, we call them stereotactic, but they are orthopedic intended use. They have an orthopedic intended use. So it depends on what the robotic surgery is doing. And it goes to a specific OHT in FDA.
That's great. Thank you so much. This question is related to our current situation, but I know that it might be out of your field of expertise. But he says that, as announced a few days ago, FDA is opening doors to at-home testing for COVID-19. Would you please clarify whether it includes both molecular tests and serological tests? If so, is it acceptable for obtaining emergency use authorization for home testing-based antibody rapid tests, lateral flow assay as well?
Well, again, I'm not on the drug and biologics, but I know that, for example, for the ventilator, we had this EUA applications. Again, we have a guidance document for EUA. Just search for the EUA guidance document from FDA on Google, and you read the EUA guidance document, and it shows you the path that you have to follow.
Thank you so much. Dr. Kazemzadeh, it's almost past 1. Would you mind to stay a little bit more with us and answer the questions? I don't know your schedule, but I know that you are a busy guy.
It's fine. I can stay for a couple of minutes.
Okay, so the next question is my question, because when I was a graduate student, like you, I'm a biomaterials tissue engineer, scientist, and I was looking and I was reading these ISO standards for biocompatibility tests, cytotoxicity, and medical device validation. But I was always wondering, how to start with any course or certificate for commercialization or medical device to be able to get the job in like regulatory agencies, either in Health Canada or in FDA. So would you give us some insights about the path that the young generation, young students, and even early career researchers or scientists can take, and if they are interested to become part of the FDA or other regulatory agencies, what path they have to take, and yeah, please comment on this.
Sure. So, well, I can talk the path I took. You don't need to have really a regulatory experience. I didn't have much of regulatory experience when I joined FDA. They train you for that. So what they need as a pre-market reviewer, if you are recruited as a pre-market reviewer, usually they need a scientific reviewer. They want someone who knows the science of that specific field. So when I got into dental, I already knew bone. I already knew all the testing in terms of biocompatibility. I was familiar with the kind of animal study they were doing. So I just need to tailor myself. And for me, the FDA was like a law school. I got into the law school and, learning about all these regulations and all these. And there are a lot of terminology and regulations that you have to catch up. So just it is important to learn all these regulations. Then you can connect them to your science, and then you become like an expert in terms of lead reviewer. Exactly. But I didn't have any much of regulatory background. But maybe I was just in the right time, in the right spot. The applications usually go through the USA jobs, and unfortunately, you have to be a US citizen to apply for it. But at the time, I was not a US citizen, so I was entered FDA through a kind of a staff fellow program. So I have seen a lot of staff fellows from ORISE as a postdoc inside FDA. They joined the pre-market after, but usually the job posting is happening on the USA jobs.
Great, thank you so much. I think there is a traditional and non-traditional way of, joining these kind of regulatory agencies. You could have related certificate or backgrounds or education on regulatory affairs, and then join or apply for the positions. Or even if you are a scientist working on different medical devices or different technologies, then you have the foundation and background, and then you have to educate yourself about the different standards, terminologies, and other things that you mentioned. So I think there are two ways to do that. So, and we have also. So the other question that I always had is that, okay, we have US FDA, we have Health Canada, European countries also, they have their own regulatory agencies, and then in East Asia, we have probably Chinese regulatory agencies. So I just want to know, what are the major differences between these agencies in terms of medical devices, or they are just the same thing, or yeah.
Yes, there are differences between regulatory agencies. That's why you see some of the devices have like CE mark, and some of the devices and still the same devices cannot get the FDA approval or FDA clearance. They have different. There are some different criteria and acceptance level in different organizations. I know about the US FDA. But if you are interested in learning about the differences between these different countries, Japanese, Canadian, European CE, or US FDA, I recommend a course which is not a course, but it is like a workshop or some sort of certificate you can get. It's called RAC. So if you look for RAC and read the book, that's a good certificate to obtain. Actually, that's in my wish list to understand the whole difference between the differences between different regulatory agencies in the world. Yeah.
Okay. And the other question is that, how long does it take for a medical device to get approved by the FDA?
Sorry?
How long does it take for a medical device to get the approval from the FDA?
Oh, I went through all that in my slides. So it depends on the kind of submission you send. Is it like 510K? Is it Denovo? Or is it PMA? Is it HDE? Or it depends on the risk classification. Different timing for each of these submissions. It depends on what your device has been classified as. PMA has different duration than Denovo and 510K.
Yeah, and another comments or question that participants have is that, is it possible to have the slides, or because we recorded the talk, so we could always you could always watch it again and get the information. But I don't know if we could get the permission from you to share the slides with the participants.
I think the YouTube would be as you said, you share them on YouTube, so they can look at the slides on there. I got a clearance for these slides, but I think the YouTube would be a better option. And the kind of Tony is putting a link for the RAC. I think that's the Tony. Yes.
Yes, thank you so much, Tony. Yeah, that's great. We will look it up. And I think that's it. And I really want to thank you, Dr. Kazemzadeh, for this informative talk. And thank you for accepting our invitation to participate to these seminars. We really got the insights how it works for the medical devices in the FDA. And I mean, I wish you all the best. And then hopefully we see you for next seminar soon for other with other subject. And
Sure. Thank you very much. Thank you for the invite. It was a pleasure attending your seminar, and I hope it was helpful.
Yeah, before I finish, I just wanted also to again kind of say that please follow us for the next talk next Wednesday at 12 o'clock, same time, same place, with Professor Jeff Karp. And he's a serial entrepreneur and professor in Harvard Medical School, and he will give us his insight about tools for accelerated medical innovation. And by this, since we don't have any other question, I would like to thank everybody to attend the seminar, and see you all next week, the same time, same place. Have a great day. Bye-bye.